Okay, one way of testing your understanding of the extracellular versus the intracellular compartment is to present to you this graphical representation. Now, the graph represents volume in the extracellular versus the intracellular and solute concentration. Now, in a steady state, the solute concentration will be the same in the extracellular versus the intracellular compartment. This was a standard graphical representation they used on step 1. They may still use this, and therefore it is worth going through and understanding how to apply this graph to the parameters that are important to us. Now, orient ourselves to the graph then. This here is the origin of the graph. So this is the origin of the 3 parameters. The first parameter I want to address here will be what is plotted on the y-axis, and that is solute concentration. So the y-axis is the solute concentration. It is the concentration of dissolved substances in the extracellular and the intracellular compartment. More specifically, it would represent the concentration of particles that cannot penetrate a cell membrane. Therefore, most of the concentration of solute here can be simply expressed as the extracellular sodium chloride concentration, or 2 times the sodium concentration. Now, we said earlier, in a normal individual under steady state conditions, the extracellular osmolarity is 300. If we consider the solid the original state, then this point here, at which this line bisects, represents an osmolarity of 300. Now, because the y-axis is solute concentration, if you conclude that the salt concentration of the extracellular compartment increases, the line shifts up the y-axis and would bisect the y-axis at a higher point. On the other hand, as shown in this figure here, if the solute concentration goes down, if the extracellular sodium chloride concentration goes down, the line would bisect the y-axis at a point closer to the origin. Therefore, this dashed line here represents a body osmolarity less than 300 milliosmolar. If the line was above the solid line, the body osmolarity would be greater than 300 milliosmolar. And don't forget, we're considering a steady state situation. Therefore, the extracellular solute concentration will be the same as the intracellular solute concentration. Now, the x-axis represents volume. We have 2 volumes to consider here. The extracellular volume is to the right of the origin right here, and the larger the extracellular volume, the further the line would bisect the y-axis then from the origin. If this is the control state situation, the point on the y-axis here would represent the volume of the extracellular compartment. Now, if the volume of this compartment increases, this line would shift to the right, and you would have an increased volume in the extracellular compartment. On the other hand, if the volume decreases then in the extracellular compartment, the line would shift to the left, it would cross the y-axis closer to the origin. Therefore, this dashed line shows a volume in the extracellular compartment less than the original state. Now, the final variable is intracellular volume, and you go to the left of the origin. Now, don't forget, 2/3 of your fluid is inside of cells, and therefore the line that bisects the x-axis here, designated intracellular volume, will be a larger volume than the extracellular fluid. Now, if the volume of the intracellular fluid increases then, the line would shift in this direction, and therefore this dashed line here represents a situation where the intracellular volume has increased. On the other hand, if you conclude that the intracellular volume decreases, the line would shift in this direction and would be closer to the origin. Now, the whole focus here is simply to interpret the graph. But the question requires you to figure out which way each parameter will change. Now, I have a specific method of working out each variable and a specific sequence, and I'll try and generalize as much as I can through here. Now, the first parameter I always determine is extracellular volume. And there's a general rule right here. That is, if you introduce fluid into the individual, if the individual is receiving any type of fluid, the extracellular volume always increases, no other possibility. On the other hand, if you have a net loss of fluid from the individual, the overall extracellular volume decreases. These are qualitative changes, so you only have to deal with directions. So just to repeat that, if you're giving a patient fluid, no matter what that fluid is, then you're giving it to the patient in the extracellular compartment, and extracellular volume always increases. On the other hand, if you have a net loss of fluid from the individual, the extracellular volume always decreases. Now, the second variable is solute concentration. And don't forget, this refers mainly to the sodium chloride concentration in the extracellular compartment. Now, simple reasoning works really well here. You just have to consider the situation and determine if the sodium chloride concentration increases or decreases in the extracellular fluid. That gives you the change in solute concentration. It also gives you the change in body osmolarity. Now, it's the last variable that is the discriminating part of the question. But you simply have to realize that intracellular volume will change based upon your conclusion of solute concentration. Now, we mentioned this earlier. If solute concentration increases, if body osmolarity increases, your cells shrink. On the other hand, if solute concentration decreases, then, if solute concentration decreases, if sodium concentration decreases, then your cells swell. So intracellular volume depends upon the conclusion then of solute concentration. Now, in the notes, we have several specific situations, and I'll give you examples of these situations, and you should be able to go through and figure out directional changes in each of these variables. Now, we'll go through some, and then you can try some on your own. Now, we're not using the graphical representation, but complete answers with explanations, including the graph, is given at the end of this section here. Now, we start with a fairly simple situation. Suppose we have a loss of isotonic fluid from the extracellular compartment. Now, this could be a simple hemorrhage. However, this is a little ambiguous because when you hemorrhage, you lose red blood cells. And technically, the interior of a red blood cell is intracellular fluid. Therefore, with a hemorrhage, you're actually losing some intracellular fluid. A better question would involve the consequences of forming isotonic urine. But more likely, they will test the acute consequences of diarrhea. Now, you have to know going into the question that diarrhea represents a loss of isotonic fluid. So you have to remember that diarrhea is the loss of isotonic fluid. Therefore, a perfect example of this situation. Now, starting with extracellular volume, you are losing fluid from the individual. Therefore, the extracellular volume has to decline. No other possibility. Now, you are losing isotonic fluid. And therefore, you should realize there is no change in solute concentration. If you're losing isotonic fluid, there is no change in the sodium chloride concentration of the extracellular fluid because you're losing equal amounts of fluid and salt. However, even though the sodium chloride concentration does not change, whole body sodium declines. You are losing sodium chloride along with the fluid, so whole body sodium declines. Now finally here, I said that the discriminating variable is intracellular volume, and that depends on solute concentration. And because the solute concentration did not change, there is no change in intracellular volume. So you simply lost a volume from the extracellular compartment, and there's no change in the concentration of dissolved substances in either the extra or the intracellular environment. Now the second example here is a very typical situation to test on step 1. They like to test, in many cases, your understanding of the loss of hypotonic fluid. Now, the very common example here is somebody who loses a lot of fluid because of sweating. They may talk about a person wandering around in the Arizona desert with very little water available, or they can talk about the consequences of exercise. You have to realize that sweat is hypotonic fluid. Going into this question, you have to know that sweat is hypotonic fluid. Now, another good example of this situation is diabetes insipidus. In diabetes insipidus, you're losing a large volume of dilute urine. Now, qualitatively, the consequences are the same as sweating. Now, being as careful as we can here, you are losing volume from the extracellular compartment. Therefore, overall, extracellular volume decreases. No other possibility. Now, if you're losing hypotonic fluid, you are losing more water than salt. And therefore, the salt concentration in the extracellular compartment increases. So because you're losing more water than salt, the salt concentration in the extracellular compartment will increase. Now, the third variable is the intracellular volume. And you should automatically say to yourself, because the salt concentration increases, cells shrink. Therefore, the intracellular volume goes down. You pull water out of cells. Now, just adding a final detail here. If you pull water out of cells, that water has to go into the extracellular compartment. Therefore, the extracellular volume increases towards normal. But it does not go back to the original level because the general point is that compensation is almost never complete. So the water you pull out of cells will increase extracellular volume towards normal, but it is still depressed from the original level. But the point is, if you go through a heavy exercise session and you lose 1 liter of extracellular volume, you will pull water out of cells. Therefore, the overall net loss in the extracellular compartment will be less than that 1 liter because you pull water out of cells. Now, that's a very important example to understand. Now, the next example here is one that they could test, and this would be for those individuals who really understand the theory here. The third example is a very discriminating situation. That is, suppose they ask you the consequences of ingesting some salt tablets. Now, the first thing to determine here is what is actually happening when you're taking in these salt tablets. And the conclusion has to be that you're simply dissolving salt in the extracellular compartment. So ingesting salt tablets is exactly the same as dissolving salt in the extracellular compartment. Now we have to be careful here with our analysis. If we look at the initial situation, if you dissolve salt in the extracellular compartment, we will assume the salt does not occupy space. So initially, there is no change in the volume of this compartment, but that's a transient conclusion. Simply dissolving salt in the extracellular compartment does not change its volume significantly because there's no additional water there. Now, going to the next variable, if you dissolve salt in the extracellular compartment, then you should be able to conclude that the salt concentration increases in that compartment. You have more salt in the body in the same volume of water, therefore the salt concentration increases. Now, because the salt concentration increases, you also should immediately conclude that your cells shrink. Therefore, the intracellular volume decreases. Now again here, you're pulling water out of the cells. If you pull water out of the cells, it will move to the extracellular compartment, and therefore, overall, you have an increase in extracellular volume. In summary then, when you take in salt tablets, you increase body osmolarity, and you induce a shift from the intracellular fluid to the extracellular fluid in terms of volume. There's a volume shift from the intracellular to the extracellular compartment. Now, as I said, that is considered a very discriminating question. Only those individuals who really understand the theory are supposed to be able to work this through. Now, the fourth and last one I'll go through here represents a situation where a person drinks 1 liter of tap water, or they could say 1 liter of distilled water. You are to assume that tap water does not contain any dissolved salts. So you get the same consequence as drinking tap water as taking in distilled water. And don't worry about the fact that they may mention taking in 1 liter. It doesn't matter the volume, because these are qualitative questions. Now, number 4 here is basically the same as water intoxication. So a person drinking tap water or distilled water has the same consequences as water intoxication. Now, again, we use our same reasoning here. If you're taking in fluid, then you're adding fluid to the extracellular compartment. Therefore, the volume in this compartment will increase overall. No other possibility. If you're taking in pure water, then the extracellular salt concentration decreases. Therefore, body osmolarity decreases, and you should immediately go to the third variable, and you can conclude if the salt concentration goes down, cells swell. Therefore, water flows from the extracellular compartment to the intracellular compartment, and therefore the extracellular volume decreases towards normal. But again, compensation is not complete, and the volume of the extracellular compartment will still be above normal. Now, just adding some quantifications we did earlier. If you drink 1 liter of pure water, you are introducing 1 liter into the extracellular compartment, but because cells swell then, the overall increase in the extracellular compartment will be less than 1 liter. So because cells swell, the extracellular volume will not change by 1 liter. The whole system is designed such that the intracellular compartment in many situations tends to buffer changes occurring in the extracellular compartment. So this huge volume in the intracellular compartment then is utilized to try and buffer volume changes in the extracellular compartment. Now, in your notes, there's several other examples. You use the same reasoning. You should go through these on your own. And as I said, the answers with explanations are at the end of the section here. You should go through and figure out the consequences of giving hypotonic saline. This is a clinical maneuver and likely to be tested. It is sometimes called giving half-normal saline. That half-normal is just hypotonic. You should also understand very well the consequences of infusing isotonic saline, because that is extremely common in the clinical situation. And finally, right here then, you should understand the consequences of infusing hypertonic saline. Now, instead of asking you about hypertonic saline, they could ask you the consequences of hypertonic mannitol. Now, it is really the same question, because mannitol is a substance that will not penetrate cell membranes. So mannitol will enter the extracellular compartment, and because it cannot penetrate cell membranes, it will contribute to the extracellular osmolarity. Now, the last situation is very unlikely to be tested on step 1, but they could ask you the consequences of primary adrenal insufficiency. Now, with primary adrenal insufficiency, you lose your mineralocorticoid activity. Therefore, you will lose volume and salt. More or less, you lose equal volumes of salt and water, and therefore then it's really an isotonic loss. But that's not a perfect description. Now, you have to understand the typical volume replacement here to get the question correct. Normally, the people will replace the volume more than they replace the salt then. So you do replace volume more than you replace the salt. So this is a very complex disorder to deal with, and it may be on what they require on step 1. Now, on the next page, then, we have tried to tabulate all the basic changes in the 5 situations they're likely to test on step 1. That is the loss of isotonic fluid, then the loss of hypotonic fluid, the gain of isotonic fluid, the gain of hypotonic fluid, and the gain of hypertonic